Friends, welcome to Biology Made Easy. We are discussing the mineral nutrition chapter, and this video is going to be the last video of this chapter where I will discuss nitrogen assimilation. When we have discussed the nitrogen metabolism, there I have told you that the nitrogen metabolism will be discussed in three parts. The first part we have discussed is a nitrogen cycle. In the second part we have discussed how this nitrogen is fixed biologically by different types of bacteria, and the third one that we will discuss in this video is the nitrogen assimilation. Now the question comes, what is this nitrogen assimilation? We have seen that in the atmosphere the nitrogen which is present cannot be directly used by the plants. Therefore that atmosphere nitrogen is fixed in the soil in the form of the ammonium compounds. Then these ammonium compounds can be converted to nitrates and nitrates by the process of nitrification. That also we have discussed in the nitrogen cycle. But all these usable forms of nitrogen, that is ammonia or ammonium ions or nitrate or nitrate, they are present in the soil. But we have to discuss how that nitrogen is assimilated by the plant body, how it utilizes the plant body. Usually, out of all these forms, the nitrate form is the mostly observed by the roots of the plant body. Then some ammonium ions are also observed. That is the most preferred one. Now, once this nitrate is observed by the roots, the plants do not know how to utilize these nitrates, because most of the metabolism of the plant body takes place in leaves. Now the nitrate will move upward, and if some ammonium ions are also observed, that will also move upward to the leaves where the metabolism mostly takes place. But how they will move? Usually the nitrates, they usually move with the help of some nitrate transporters and through xylem only. Similarly, if ammonium ions are observed, then they also move through ammonium transporters and again through xylem only. That means through xylem, the ammonium ions or the nitrate ions which are absorbed by the soil, they will be translocated to the upper part of the plant body, that means to the leaves, where it will be assimilated. So how this assimilation is taking place, let us discuss step by step. Now once the nitrate ions reach the leaves, how it will be assimilated? It will be assimilated in different steps. Now in the step number 1, the nitrate which has now reached the leaves, it will be converted to nitrite. And where this will take place? This will take place in the cytosol of leaf cells. And for this reaction, conversion of nitrate to nitrate, the enzyme is nitrate reductase. And this nitrate reductase is cofactor is molybdenum. That means the molybdenum is required for its activity, is a cofactor, and this nitrate is converted to nitrite only in the cytosol of the leaf cells. And another very important thing is this enzyme is an inducible enzyme. It is an inducible enzyme. Now what is the meaning? That means only when this nitrate is available in this cell, then only the nitrate reductase will be formed, otherwise not. It is inducible. It will found that the nitrate is available in soil, then that will be formed and immediately will be converted to nitrate. Otherwise, in the rest of the time, it is not there. That is very important. So this is step 1. In step 2, what happens? In step 2, now this nitrate will be converted to ammonia. This will be converted to ammonia, or we can tell that even also ammonium ions. It is converted to ammonia. And this reaction takes place in the presence of the enzyme nitrite reductase. Nitrite reductase, whose cofactors are usually iron and copper, not molybdenum. These are very important from examination point of view. Here the molybdenum is cofactor and here the iron and copper are the factors. And when this reaction is taking place, it also requires particularly, because this is a reduction. When this is the nitrate is converted to nitrate, so there is reduction. So here it is reduced. Nitrate is reduced to ammonia, and the reducing agent is NADPH, the reducing substance. And this reaction takes place inside the chloroplast, and it always takes place in the presence of light. Why in presence of light? Because when a light falls on the chloroplast, there will be photolysis of water, and during that photolysis of water, this NADPH is produced, which will reduce this nitrate to ammonia. So you see, this reaction takes place in the cytosol, while this reaction takes place in the chloroplast. It is not depending upon light, but this is light dependent reaction. This is nitrate reductase, this is nitrate reductase, and here the cofactors are molybdenum, here the cofactors are iron and copper. Now, inside the chloroplast, the ammonium ions or ammonia is formed. Now, this ammonia can take part in the formation of amino acids. So, the next is step 3. So, in this step 3, we will discuss how the amino acids are synthesized. Step 3 is amino acid synthesis. And you know that for proteins there are 20 different types of amino acids. How these different amino acids are synthesized? So they are synthesized by 2 different processes. One is reductiveamination. Reductiveamination. In this reductiveamination, the alpha ketoglutaric acid, this is a 5 carbonic compound organic acid and one of the intermediates of Krebs cycle. So this alpha ketoglutaric acid, usually it combines with ammonia, and it usually gives rise to, it can combine with the ammonium ions and along with the NADPH, and it will give rise to glutamic acid or glutamate, and this is the amino acid, this is the organic acid. So now it is converted to this, and now the air will get one molecule of water and NADP is liberated. Now, why it is known as reductive amination? Because in the organic acid, there is a carboxyl group. So this carboxyl group is reduced, and there is amination of the organic acid. That means the reduction of the carboxyl group and also the amination of the organic acid. That is why it is known as reductive amination. And this reaction takes place in the presence of the enzyme glutamate dehydrogenase. So now one of the amino acid, that is glutamic acid, is formed as a result of reductive amination. In this manner, another two amino acids also can be formed, that is the oxaloacetic acid. The oxaloacetic acid can be converted to aspartic acid, and the pyruvic acid can be converted to alanine. Maximum these three amino acids can be formed by reductive amination, not the remaining 17. Now the remaining 17 amino acids are formed in the next step, that is known as transamination. So in the second step of this amino acid synthesis is transamination. So the step B, transamination. In transamination, what happens? Suppose let us take glutamic acid, and glutamic acid was an amino acid, and let us it combines with another. Suppose let us take oxaloacetic acid. This is organic acid. That is, this is organic acid. This is amino acid. Now, in presence of the enzyme transaminase, what will happen? The amino group will be shifted to this. So this will be converted to corresponding amino acid, and the keto group will be transferred to this group. That means now this glutamic acid will be alpha ketoglutaric. It will be alpha ketoglutaric acid. That means this is now organic acid. This will be converted to the corresponding amino acid. This is aspartic acid. This is now amino acid. This is known as a transamination. Now, if we can write it also like this. Suppose this is one amino acid. Now, this one amino acid, and let it combine with another organic acid. This is organic. This is amino acid, and this is organic acid. We can write like this, or also we can write, if we will write it as CO minus, then in that case this will be NH3. In that case it will be NH3 plus. And here also we can change it. Now what is happening, you see. Now how the amino group and keto groups are exchanged. Now this will be R, this will be C, this will be H, and now this will be double bond O, and this is the COOH. That means this amino acid is now converted to organic acid. And the second one will be R, C, H, COOH, NH2. Or you can write this as CO minus, and this will be NH3 plus. That means now the amino acid is converted to organic acid, and organic acid converted to amino acid. So in this way, the remaining 17 amino acids are formed. So now these are the two processes. One is the reductive amination, in which glutamic acid is most formed, and from this glutamic acid the remaining acids can be formed by the second process, that is transamination. And sometimes the aspartic acid and alanine, they can be directly formed by the reductive amination, or they can also be formed through this method also, transamination. So now all the 20 amino acids are formed by this process. Now you see, the nitrogen which has been fixed in the soil by means of biological nitrogen fixation, that is absorbed in the form of nitrate and ammonium ions, and now with the help of the nitrate reductase, that is again converted to ammonia, because ammonia or ammonium ions can be directly used, and now they are attached with the organic acid to give rise to corresponding amino acids. Now these amino acids will combine together. there will be polymerization, and that will give rise to proteins. This is the method how they are formed. But in some plants, what happens? The amino acid, in the amino acid, instead of a single amino group, two amino groups are attached. So that gives rise to amides. But that doesn't take place for all amino acids, only for few amino acids that take place. So you can write step 4, that is formation of amides. So in case of amides, there is more than one amino group. So here you see one amino group, this is CHNH2 and this is COH. This is amino acid. But when it will amide, there will be more than one. Where is the second one attached? The second one is attached to this one OH group is replaced, and here this is NH2. That means now there are two amino groups. So this is known as amides. And the common two amides, one is glutamine and the other is asparagine. These two, and for their synthesis it requires glutamine synthetase and it requires asparagine synthetase. For them, this is asparagine synthetase and this one requires glutamine synthetase. So these are amides. And in case of soybean, these glutamine and asparagine, these are amides, that means they contain more nitrogen and usually they are translocated through the xylem. Then step 5, another exception is there. In case of soybean, usually the amino acids are converted to urates. They are urates, and these urates, they are usually translocated to different parts through the xylem. And since there is more than one amino group, this nitrogen by carbon ratio is more. These are all important from examination point of view. So you see how the nitrogen is fixed in the soil, and that is assimilated, and finally it is incorporated in the form of amino acids and also in the form of proteins. So this is how the nitrogen is assimilated. So with this, the entire mineral nutrition chapter is complete. But let us discuss questions. So one question we can take. Nitrite reductase requires dash as cofactors. A choice molybdenum, iron. B choice iron, copper. C choice copper, molybdenum. And D choice molybdenum, copper, iron. Then the second question. In the second question you can write during which conversion reduction occurs. A step when nitrate is converted to nitrite. B choice nitrite is converted to ammonia. C choice both steps. And D choice none of them. Very good questions. These are the type of standard questions asked usually all in all national type of examinations. Thank you very much for watching it. God bless you. Have a good day.